So I want to thank the organizers for the opportunity to talk about my work. It's kind of an earlier version of what John Flannery was talking about. We're using an optogenetic approach where we're targeting it to the bipolar cells. The work I'm going to be talking about today is specifically with channel rhodopsin 2, and a lot of this work was done in collaboration with Bill Houseworth. I'll skip over this, but the main motivation is that there are clearly a number of people that suffer from blindness from photoreceptor diseases, specifically retinitis pigmentosa and age-related macular degeneration, and particularly the dry form. And this number is continually increasing as people are able to live longer with improvements in other medical technologies. And we expect this to increase by 50% by 2020. I think we all know these numbers. And previously I've been saying that there were currently no FDA-approved treatments. As some of you know, or maybe many of you know, Second Sight Medical Products has created a epiretinal prosthesis that stimulates the ganglion cell side of the retina, and that has been approved for market, and it should be on the market, I think, in the middle of this year, if not later in the year. But one of the problems with this is an earlier version of the electrode array. Is there a pointer? I can use that. These are 500 micron electrodes, and each ganglion cell is somewhere in the range of about 10 microns in diameter. So each of these electrodes is stimulating multiple cells all at the same time. And ganglion cells, there's probably 20 different types of ganglion cells transmitting 20 different types of information. And so you're stimulating these ganglion cells not indiscriminately, so you're stimulating on versus off at the same time. And in addition, which you can't really see here in this fundus, is fibers of passage. So the axons form bundles and go towards the optic disc. And what you find in the patients, so I worked with these patients for about 5 years during my PhD. And one of the first questions I asked is, well, what do you see when we stimulate with electrical current? And so this is in one patient. I stimulated 2 electrodes, and this is about 2 millimeters apart. And I asked them to describe what they saw, and then I just simply drew it out. And this is repeatable. I did this multiple times over multiple days, and this person saw a line here and then a little dot up here. And then if I stimulated 4 electrodes, they saw several lines in different orientations. So this is suggestive of fairly indiscriminate activation, in particular the fibers of passage. So this work I'm going to describe now is motivated by this fact. Could we apply some technique to be more specific in how we activate the retina? So in about 2007, I found out about optogenetics and spoke with Ed Boyden about the opportunity of making other cells in the eye light sensitive. So in the normal case, you have light going into the eye, activating the photoreceptors, and in case of RP and AMD, you lose these photoreceptors, so there's no way for the eye to capture light. So what we're trying to do is simply introduce, specifically, expression of channel rhodopsin 2 into the on-bipolar cells.
So as to optimize its photosynthesis.
Oops.
DNA.
So the way we were taking a gene from an algae, it's channel rhodopsin 2. It's about like 1 kb. We introduce it using an adeno-associated viral vector into neurons, and the protein is then expressed on the surface of the cell. These are transmembrane proteins. They go through a conformational change when activated with light, allowing cations to flow into the cell. Essentially, now you're able to depolarize neurons with light. And so the way, similar to John, the way we're introducing this into the retina and being specific to the bipolar cells, we're using the GRM6 enhancer sequence to restrict the expression to the on-bipolar cells. There's a simian virus 40 promoter, it's a core promoter, and then we're expressing channel rhodopsin 2 with EGFP fused to the channel rhodopsin 2. So anywhere channel rhodopsin 2 is expressed, we can localize it with the GFP signal. So one of the questions that we had too, and we looked at this after the fact because we were actually surprised to see expression. I think many people here, again, are familiar with Robert Mark's work. What happens in the bipolar cells after the photoreceptors degenerate, the bipolar cell dendrites retract. And this is where actually much of the metabotropic glutamate receptor 6 is expressed. And that is controlled by GRM6. And then once the dendrites retract, you see some translocation of the expression of metabotrophic glutamate receptor 6, but it's very little. So one of the things that we looked at is we actually did a full transcriptome of the RD10 retina compared to wild type, and we found that the actual transcription of GRM6, or metabotrophic glutamate receptor 6, is actually upregulated by 3 times, but the expression is still much, much less. So when you look at the expression of a protein, that doesn't necessarily mean that the transcription is going to be lower. So in our case, we find that that transcription is actually highly upregulated, which is a good thing for us, since we're using that enhancer sequence, the GRM6 enhancer sequence, to target the expression. And then we're also using a AAV8 with a tyrosine capsid mutated virus. I think many people are familiar with that, but just to tell people that don't, on the surface, there are these tyrosine residues that can become phosphorylated that mark them for degradation. So when you do the injection, many of the viral particles can be degenerated. So if you remove the tyrosine residues, even one of them, you can actually improve the transduction efficiency dramatically. So that's what we've used here, is just a single tyrosine mutation to a phenylalanine. This is an RD10 retina. This is looking at the inner nuclear layer, the bipolar cell layer. The photoreceptor layer would be up on top. It's essentially absent. The DAPI is showing you where the cell bodies are. PKC alpha is a marker for rod bipolar cells, a type of ON bipolar cell. There's also the ON cone bipolar cells. And then you can see here at the top of the inner nuclear layer, you can see green and then also yellow and red. And then down below, this is the inner plexiform layer, which is where the bipolar cells are synapsing onto the ganglion cells. You can see 2 stripes. One is this green stripe up on top, where the ON cone bipolar cells are synapsing onto the ganglion cells. It's very distinct. And then right below it is the rod bipolar cells synapsing onto the ganglion cells. So you see that both cell types are actually synapsing onto the ganglion cells and expressing channelrhodopsin 2. And it appears that the off bipolar cells are not expressing any sort of channelrhodopsin 2. So this is an indication that we're being very precise in where we're expressing our protein. And then down below, this is just a further zoomed in image showing that our expression is nicely localized to the surface of the protein, not in the cytoplasm. And one other question that we had is, how long does this expression last? This is just taken from the previous slide. This is showing expression 10 weeks post-injection. Incidentally, for those people doing gene therapy, we didn't see any expression essentially up to 2 weeks and even 4 to 6 weeks. About 8 weeks, we started to see some expression, but at 10 weeks, we really saw good, robust expression. I think this has to do with using a cell-specific promoter. But then also at 10 months, we saw good, robust expression. So the expression is well maintained. These were done in animals that were approximately 2 to 4 months of age. One question that we received is, well, what happens when you try to introduce this virus in a much older animal? So we actually took an animal that was 20 months of age, an RD10 animal, 20 months of age, which is about 70 to 80-year-old person equivalent, and injected our therapy. And this, we did get expression. And I can show that image. I don't have that here. But we did get expression in the inner retina, in the ON bipolar cells, although the retina is pretty messy. So we also looked at physiological responses in terms of efficacy. These, on the first column here, is channelrhodopsin 2 expressed in the ON bipolar cells without photoreceptors in the RD10 animal. Then next to it is the RD10 untreated, and then the wild type. And so we presented a 3-second burst of blue light. And in the treated animal, we did see a good, robust response, physiological response. This is an MEA recording. This is the retina is taken out. It's ex vivo. And then in the untreated animal, we saw an absence of response, physiological response. And then again, of course, in the wild type, we saw a response again. And we did do some blocking to make sure that this wasn't due to any sort of residual photoreceptors, and that we do indeed find that this is due to intrinsic light sensitivity in the ON bipolar cells. And then also we looked at the behavioral task, where an animal was trying to find a platform that was tethered to a light source. It was navigating to 6 choices. And after 2 weeks of training, wild-type animals, more often than not, got this correct in about a couple of seconds. So the amount of time it took to find that platform was some indication of their visual ability to find that platform. And if you look at the untreated animals, these are RD1, RD16s, and rhodopsin knockouts. We looked at 3 different models. Again, we trained them for 2 weeks. they know that there's a platform there. We can actually navigate them with a little stick, so they know that there's a way to get out of the maze. But over time, they develop a strategy which just involves tracking along the walls. So they just whisk along the walls until they actually are able to find that platform, which obviously takes quite a bit longer than, say, just visual information, navigating to the escape. So these are the same model animals then with the treatment, with the channelrhodopsin 2 expression in the all-bipolar cells. As you can see, they behave much more like the wild types. And one thing to note, if you look at their heads, they'll actually orient towards the light source, showing that they actually do maybe see the light in the periphery and actually give them some improved navigation ability. One thing I do want to say about these animals is that this task in particular doesn't say much about visual acuity, but it does give you a lot of information about detection and navigation. But it does take a fair amount of light. Channelrhodopsin 2, as you know from the previous talks, is actually fairly insensitive. We found that about 10 to the 14 to 10 to the 15 photons per centimeter squared was sufficient to drive this behavior. Again, as many people have said, that's probably the equivalent of being outside on a bright sunny day in Miami. We also found that this behavior is well maintained 10 months post-injection. So not only do we see expression 10 months post-injection, we see the behavior is well maintained. We looked at a number of different safety endpoints, including biodistribution, immunogenicity, and inflammation. And we looked at this in 2 lighting conditions. One's in normal lighting conditions, 12 hours on, 12 hours off. And in that case, we didn't see any changes in GFAP expression, nor did we see any infiltration of immune cells with the CD45 marker. And incidentally, at 10 weeks with high intensity light exposure, basically those same light projectors that we were using for the behavior rig up against the cage, 12 hours on, 12 hours off for 10 weeks, we didn't see any immune cell infiltration nor any increase in GFAP relative to the disease model. Now, the disease model does show some GFAP expression, as you can see in the control up here. So I think this, in summary, the therapy is ready for development. We have spoken to the FDA with a pre-IND meeting. I actually agree with what Thomas said. I think it's maybe a mixed bag to speak with the FDA too early. Our biodistribution is nicely localized to the eye. We don't see that this is immunogenic or no signs of inflammation. And we have looked at this a little bit in primates through Ed Boyden, where he did some injections in cortex, and he found that channelrhodopsin 2 expression actually isn't causing any sort of inflammatory response there, even after a year's time. And now we're currently looking at 2 animal models outside of mouse, looking at a pig model of retinitis pigmentosa and also a rabbit model, which is a rhodopsin mutation with Robert Mark. So I just want to talk a little bit about what we're doing now. We're doing some optimization work where we're trying to look at other options that may be more sensitive. Channelrhodopsin 2, of course, takes a lot of light. We're looking at other channelrhodopsins that have better biophysical properties, and also G-coupled proteins as well. So one of the things that we've been doing with the mice recently is using an endoscope for the injections. This is done with Nate Roybal at UCLA. This is the disease RD10 retina at 6 months. You can see good images of the back of the retina, changes in pigmentation. And this is a pre and post injection. This is done with fluorescein, a nice subretinal bleb. And I just want to show you a quick video of that. It's taken with, I apologize, it's taken with an iPhone. I'm actually plunging the actual injector at the same time while holding the iPhone on the screen, but you can see that the needle is going through the sclera right up into the retina. And then in a moment, you'll be able to see that the subretinal bleb is nicely formed, fluorescein. So the nice thing about this technique is it actually provides a lot of confidence about how well your subretinal injections took. And so you can write detailed information about how good the injections were, and then compare that to your end results. So the endoscopic technique helps a lot. As I mentioned, this does take a lot of light, so in terms of what we can expect, it might be that if we were to treat a patient, they may see moonlit type experience out on a very sunny, bright day. In terms of visual acuity, this is a bit hand-waving. I think under the best conditions, we're probably looking at 7 cycles per degree, which is about 20/80. And if we actually start to think about retinal reorganization and many other things in terms of changes in contrast sensitivity, this is probably an order of magnitude worse. So we're probably looking at twice the visual acuity of a mouse, which is still actually quite good, but that's about 2,800. Quite good for someone who's never, they can no longer see. I should be clear on that. In terms of navigation, I simply have no idea. I think that's an open question about how motion processing will be accomplished in these folks. This was a lot of work that was done at USC and also with the company US Neuroscience. I'm just as a matter of disclosure, the chief science officer and co-founder of the company. I founded this with Ed Boyden at MIT and then also Ben Mateo, who's in the Bay Area. And I just want to acknowledge a number of people that were working with us on this project. Some of this early work, again, was done with Bill Hauswirth and Boyden, and we're starting a number of different collaborations with Brian Jones and Robert Mark, and also Ed Stone and Elliot Sun at University of Iowa. And I also just want to recognize a number of funding sources that supported this work. Thank you.
I want to thank the Hope for Vision group and all the colleagues here for this absolutely spectacular meeting. As a sort of a clinical person, I thought I would approach the clinical issues that I worry about, which is with this kind of work going on, and it's really amazing what's going on at the moment, as presented by my colleagues. I think there's a need at this point to decide who are the candidates. I know that candidates are already being involved in trials, but it may be really worthwhile to start thinking a little clearly about inclusion and some criteria for benefit from some of these novel therapies. So I want to thank my collaborators. So as we all know, this is the retina, and this is the area of photoreceptors that are dysfunctional or degenerative or maldeveloped, and which then don't survive. And frankly, this is our problem zone. So optogenetics, or actually stem cells, or actually electronic prosthesis, or many of the topics that were brought up today, are fields that really are gaining in knowledge, and actually over the optogenetic field is over many decades. And the purpose, at least for some of this, is noble and scientific, and has been really beneficial for all of us to express light-activated proteins in specific neurons, and that's brought neuroscience forward. But it's also been considered, as we've heard, for biomedical uses, and that's the purpose of this meeting. So we've heard from acknowledged experts in the field over the day, and here's some of their papers, and I won't go through that again, but the field has moved forward, and there's more to do, though. So who is an appropriate candidate for this type of procedure? And I don't think that's a trivial question, at least for me, because I'd like to know that the patients I advise or advocate for to have some kind of treatment like this actually might qualify. So we've sort of been down this pathway before of trying to figure out retinal criteria for some sort of therapy. It was actually a gene replacement therapy, augmentation therapy. And so in that case, we, back in 2005, we worked very hard to search at the time when retinal generations were, we were working on using optical coherence tomography, the wonders of which we all know, to determine photoreceptor content of retinas. And so in the RP65 patients, we identified photoreceptors and then felt more secure in mapping them and then delivering treatment targeted at those photoreceptors rather than just sort of a routine injection somewhere. So for, let's go back to optogenetics as an example. The criteria are obviously very different, and then RP65, here we're actually looking for absence of photoreceptors. And after a quick breeze through methodology, and I will breeze not to take too much more time, I thought we'd quickly just talk about a few different patient groups, again in brief, as examples of who would be patients that we would actually recommend for this and who obviously wouldn't be. So we need measurements, and I think measurements are always useful. I saw measurements in all the scientific slides, and I think that has to also apply clinically. We have to obviously know what the visual function is. We're not putting 20 people into this situation, and we should have some retinal and post-retinal structure. We've been collaborating successfully, I believe, with Jeff Aguirre, who is a wonderful neurologist who has a fondness for brain imaging and MRI and structural and functional MRI, and we've looked at various forms of retinal degenerations with Jeff, so I won't go into that. So now we'll go back to our famous optical coherence tomography, which has been of tremendous value to all of the retina folks. We're used to using these color maps to make clinical diagnoses and make decisions about patients in the clinic, but you can display it any of a number of ways in grayscale or however you want to display it, but the basics that you need are to know some sort of have some understanding of what the measurements are in these sublaminae. So the layers are hyporeflective and hyperreflective. This is a reflectance technique, and so over the years, and starting with all the original studies back in 90s, we've tried, we and others, and a lot of others, have tried to identify whether these layers have anything to do with the retina as we know it, and I think we're pretty happy now with what these layers mean. And for this particular work, we're looking at the, we're going back here, we're looking at the photoreceptor layer, we're looking at, actually we're looking at the nerve fiber layer and the ganglion cell layer, and those are the ones that we're interested in if we're thinking about, say, for example, a retinal ganglion cell therapy. Methodologically, I'll start showing you slides that will, this is a normal, these are all normals, and so this is a normal cross-section colorized for your pleasure of the outer nuclear layer or photoreceptor layer, the ganglion cell layer. So we see orange. Orange is good. Blue is even better, but blue tends to disappear in the diseases we're working on. And the nerve fiber layer, not the National Football League, is up there in the corner. We tend to look at normal ranges, and gray is normal, and this is ONL normal, this is ganglion cell normal, this is a map, color scale map from an OCT. This is borrowed from a variety of different sources, including Christine Curcio's ganglion cell topography paper. But we can see here that where the ganglion cells dip in the center, right here, the photoreceptors peak in the center, and there's a sort of a displacement of ganglion cells around the parafoveal region to accommodate development and foveal development and known anatomy. So this slide has been shown more often than I imagined today. But it kind of changed me a bit, I think I understand the principles that if we're dealing in primates, and the human is a primate, and the human has a fovea, and these monkeys have phobias, these macaques have phobias, that we may at least at first, pardon the word shot, injection, be really only serving a ring with a few other cells around, a ring of parafoveal ganglion cells, assuming we're going for ganglion cells. We heard about bipolar cells. We know about the attempts to do put optogenetics in cone cells. But all in all, let's think ganglion cells. That's sort of almost at the practical edge of reality here. And so parafoveal cells, well, that's not too bad. If we can only get, we don't want to rip up this retina. We don't want to peel these membranes. I have patients that have looked like they've gone through world wars after membrane peeling of their epiretinal membranes in an RPI. So we don't want to do that sort of thing. We don't want to pour Drano in here to punch holes. We really have to, I think, deal with what reality tells us there is. And right now, at least my understanding of reality is that we're dealing with this ring. So we have to worry about the ganglion cells that are, and the axons from these ganglion cells going to the optic nerve, and maybe we can get some help for people in that region. So these are 3 methods that we use to quantify ganglion cells, not ganglion cells, but we talked about ganglion cells, but nerve fiber layer. So either cross-sectional cuts like OCTs do, topographical maps as we do and other people do, and polar plots around the optic nerve, which has become conventional. So briefly, I wanted to talk about 4 different diseases very briefly. 2 forms of RP and 2 forms of Leber congenital amaurosis. As has been mentioned already, there were 2 groups actually. This stellar group in Miami and a group I worked with for many years, and in Jerusalem that's virtually simultaneously discovered that autosomal Jewish heritage homozygous founder mutation was actually causing a substantial amount of retinitis pigmentosa in Ashkenazi Jews, sorry. Ashkenazi, not autosomal. Anyway, so DHDDS, as people will tell you later, is a form of our ARRP. So let's look at some of the patients. Okay, so so here are some patients. This is a 19-year-old man, 24-year-old, 34, 37, 30-year-old. And the major message here is that we're losing outer nuclear layer. We're losing photoreceptors to the point here where there's still some reasonable visual acuity, 2200, 2100. Here there's sort of more like hand motions, light perception in one eye. And so the question becomes, what's left? Do we have sufficient ganglion cells? Do we have sufficient nerve fiber layer? Can we just assume that, or can we actually prove that? And I think we should prove it. So we looked at the photoreceptor plots and the ganglion cell plots, and there is parafoveal ganglion cells. They get a little thick here, and as shown here. And we actually sampled many loci around the parafoveal and toward the optic nerve head, and did parafoveal neural plots, polar plots of thickness. So these are all thickness measurements. So all in all, really the person of concern here, the 30-year-old with very limited visual acuity and perception anywhere in his field, is the person that we would consider as a candidate for with the DHDDS RP for this type of approach. Okay, how about CRB1 RP? So what is CRB1 RP? Well, it's a long history, which I won't bore you with. A lot of luminaries along the way making observations in Holland and John Heckenlively in California at the time, and Lottery and Stone and Hollander further along the way that moved the field forward. But this is sort of RP12, which was popularized as a form of RP based on this large Netherlands pedigree. And it was a little unusual. It wasn't exactly what RP was all about, but it was certainly qualified as RP. And so our CRB1 is actually a very well-researched article going back to Drosophila, and it has considerable notoriety from various groups that have explored it very widely. Some have even used the CRB1 mutant mouse as a way to actually, because of its ELM problems, it's a Mueller cell photoreceptor connectivity issue, to use it to move transplanted photoreceptors into degenerating retina. But all in all, it's well investigated. Now, we looked at the patients, and we came to the conclusion that it was quite a very dysplastic retina. Okay, so here we're looking at maps, and here's a normal map of photoreceptors, and these were just sort of warm is big, and red is big, and red is bigger, and so these were very thick retinas full of little pieces of possibly cells migrating through the retina, and so it's a complex retina. But all in all, here you could have in people who have complained about visual acuity loss and field loss and night blindness, maybe in their second decade of life, who started out life what they thought was normal. So this is the RP version. So the bottom line here is that here we have people with 20/20 vision, 20/50 vision, 2100. This little crumb is 2100's worth. And here's a light perception, barely person in there at age 50. And we looked at the ganglion cells. Here's the decrement in outer nuclear layer. The ganglion cells around the fovea are there. It gets awfully thick, but they're either normal or thick. Okay, we can debate forever about what the thickness is all about, and the polar plots actually confirm that. So in this group, this person here is a candidate for sure. I couldn't have told you that by just looking with an ophthalmoscope. So CRB1 LCA. So what's LCA? So LCA is the infantile or childhood version of RP. We won't go through that too much, except the fact there's many genetic defects associated with the LCA. And the ones that I just want to briefly touch on today are the CRB1 and CEP290. And many